are naked and some have an envelope which is made up of lipid. Some viruses can be made up of RNA or DNA, which can either be single-stranded or double-stranded. And then I talked about the problems we have in terms of classifying viruses, because there is no set system, even though with genotyping now, with sequencing, we have a better idea, or we're starting to get more structured ways of classifying viruses. But nevertheless, the popular mechanism is still the old traditional mechanism of giving a name based on the disease it causes, where it was discovered, or whatever. And in this lecture, I will talk about the different strategies that different viruses use to cause disease. So the objectives of this lecture is to talk about the different mechanisms of pathogenesis and transmission, to talk about the special group of viruses which can actually directly cause cancer because they contain within their genome the ability to express various proteins, which we call oncoproteins, which when introduced into a cell can disrupt various aspects of the cell cycle regulatory system, as I told you in the immunology lecture on tumor immunology. And then finally, I'll talk about the techniques that we use to diagnose viruses that you can see from this current COVID-19 pandemic how problematic this is, because the easiest way, the cheapest way is to look at antibodies. But in the case of COVID-19, it doesn't seem like there is a prolonged antibody response, that the antibody production only lasts for 6 months, which is very unusual. So these rules are constantly being changed with each new discovery or each new virus that we come across. So first of all, I'm sure you know this already, but you need to be aware of this terminology. So we talk about viruses being pathogenic, which refers to the severity of the disease. So coronavirus, the virus that causes the common cold, is less pathogenic than something like Ebola virus, which causes hemorrhagic fever. When we use the term virulence, it's to compare the degree of pathogenicity between two different strains of the same family of viruses. So, for instance, in the case of human immunodeficiency virus or HIV, which causes AIDS, there are two major strains, HIV-1 and HIV-2. HIV-1 is the one which is more virulent because it is the one that has higher chance of causing AIDS and is more transmissible compared to HIV-2, which is more restricted in West Africa. A lot of viruses can be subclinical. So it means that if you catch the virus, you can feel unwell, but not sick enough to have to go to the doctor for treatment. And then again, if we look back at COVID-19, we can see that we have a whole range or a whole spectrum of pathogenicity. So we have the elderly, the over 65s, the diabetics, the ones with hypertension, who get the most severe disease. This is clinical. The young kids under 15 years old, or teenagers, they get asymptomatic. So they may be infected, they may produce antibodies, but they don't have any effects from this virus at all. So again, we're seeing that many viruses can have very different effects or different pathogenicities depending on what we don't know. I mean, obviously, patients with predisposing conditions, you'd think that they would be more sensitive to the effects of virus infection. But in terms of age, in terms of sex, some viruses we know affect, and the same with COVID-19, that more males suffer from severe disease compared to females. What is the connection? We don't know. And then, as I said, that with the pathogenicity of these virus infections, the pathogenicity isn't only due to the virus infection itself, because many of these viruses don't actually cause what we call cytopathic effects. They don't actually kill the cells that they infect. But what they do is they cause these infected cells to express virus proteins on the surface, which generates an immune response. The inflammatory cytokines, the cell damage results in the symptoms that you experience. So then, because these viruses are packets of genetic information which rely on the cell's machinery, the ribosomes, the DNA polymerases, and whatever, to make more copies of themselves, the replication of viruses is very inefficient in that during an infection cycle, let's say only about 40% of the new viruses that are made will be infectious. The rest are what we call defective particles. So they will contain no genetic material in them. They'll just be empty capsids. And we exploit this by using this to make vaccines. So one of the ways that we generate, for instance, one of the first vaccines that was made against poliovirus was to isolate the virus that infects humans and then culture them in another species, in this case, monkey cells. And after 50 to 100 generations in this different species, the virus eventually evolves or mutates such that it loses all of its pathogenic genes for humans and becomes adapted to grow in monkey cells. So that when you put it back into humans, it's no longer dangerous to the human. So this is a mechanism for creating what we call attenuated viruses or inactivated viruses. I'll talk about that more in an immunology lecture when I talk about development of vaccines. Then in the laboratory, when we look at the effects of virus infections in cells, certain viruses have different patterns of how they affect the cell. They can create the cells to burst by necrosis, or they can cause what we call inclusion bodies, which if you look under the microscope, you can see big fat granules which represent aggregates of virus capsids being churned out before they're being processed, and these can just clog up the whole cell. So these characteristic effects is one way that we use to identify what type of viruses are infecting a person. This is not commonly used because it's very laborious. You need to grow cells, normal healthy cells in the laboratory. You need to isolate blood samples or whatever body fluids from these patients, treat them with onto the cells, wait a couple of days, and then see what happens. This is one of the ways that the COVID-19 was identified, by taking body specimens and putting them onto bronchial, cultured bronchial cells. So this is an example of the so-called defective interfering particles. So you can see here, this is Epstein-Barr virus, which is a herpes virus which causes B-cell lymphomas. And you can see one of these defective empty capsids where there's nothing inside. It's based on electron microscopy, whereas in this one, you can see the circular DNA of the virus particle. Similarly, with hepatitis B virus, hepatitis B codes for 5 different proteins. One of its most abundant proteins is the so-called surface antigen. And so if you look in the blood of patients infected with hepatitis B, under the electron microscope, you can see much greater numbers of these so-called filamentous particles. These are just empty proteins. These are just protein aggregates, whereas these are the actual infectious virus particles. So it means that during the replication process, these viruses are churning out tons and tons of these proteins, which create these so-called virus-like particles, but they're non-infectious. And only a small proportion of the products that come out of this replication process are actually infectious virus. And again, in this way, this was one of the first preparations of vaccine for hepatitis, because what scientists did was to purify the infectious particles away from these surface antigen particles, and then use those surface antigen particles as the vaccine. Now, that's a very dangerous procedure because you have to make sure that you don't have any contaminants within that vaccine with infectious particles. So a more safer way now is that we can take out the gene from this virus that codes for these surface proteins and then insert them into yeast cells and get the yeast to make these surface proteins. So this is the source of the hepatitis vaccine. But that comes on to another point. As I said, when you are creating a vaccine out of these purified particles, because in the case of hepatitis B, most of the particles that you see in a replication process are non-infectious. So that comes to another aspect of viruses. If we compare or look at the virulence or the pathogenesis of a virus, what is the infectious dose? How many virus particles do you need to infect a person to cause disease? And that can vary from 1 or 10 virus particles, as in the case of Ebola virus, to a couple of thousand particles, as in the case of influenza or the cold virus. So this is a reflection either of the number of non-infectious particles within that mass, if you're sneezing or excreting it through the fecal oral route, or how potent that virus is in terms of locking onto a target cell and then generating whatever changes it does to the cell. So that's another thing to consider with regard to the pathogenicity of viruses, is this infectious dose. How many particles does it take to cause disease? Because that relates to the transmissibility as well. And this comes down to, again, related to the COVID-19, there's been this constant talk, how protective are masks? Does social distancing work? How infectious are surfaces where the virus has been gathered, and so on. So with different viruses have different patterns of replication and disease. So the first is in the case of, say, influenza, which I'm sure you've all caught before, or colds, which are acute infections. So basically, you catch the virus starts replicating in your bronchial tissues, you generate an immune response, you create cytokines which make you sneeze, have runny noses, give you a headache. That immune response manages to kill off all of those virus-infected cells. You develop productive antibodies, and that happens in the space of 10 days to 2 weeks. That is an acute infection. You have a flare-up of virus, immune response comes in, interferons get made, the virus goes down. Then you have viruses which, just like in a chronic inflammation, have these cycles where the virus can infect cells, get a burst of replication activity, get an immune response to start killing them, and some cells still retain virus in them, and they survive this immune response. And then after a few months, they can come up again. You get another wave of killing, and then they'll go down. So this is a persistent or chronic type of infection. An example of this is with the hepatitis viruses, hepatitis B and C, which have a high rate of liver cancer associated with these two virus infections, particularly in these chronic infections, because these viruses themselves don't kill the cells that they infect, but they generate an immune response against them. And so after 30 or 40 years of these constant cycles of immune attack, replacement of hepatocytes to replace the ones that got killed by the immune system, you get, with this constant regeneration of hepatocytes, you start to get mutations in the newly generated liver cells, which can be tumorigenic and grow into liver cancer. So it's a type of type 4 hypersensitivity reaction, chronic, due to the virus. That is one mechanism. So that is what we still have arguments amongst virologists between what is persistent and what is chronic, because they more or less mean the same thing. But with persistent, some virologists refer to that in something like measles, where when the measles virus infects cells and generates an immune system to kill most of those infected cells, the virus still persists or remains in significant numbers of cells to create low level of infectious replication. And again, after 10 or so years, you can then get a neurological damage as a result of measles infection. And this is the result of immune attack, like a cross reaction, almost like molecular mimicry against nerve cells. So this is a persistent infection where the acute infection got controlled by the immune response, but the virus hasn't been completely eradicated, so you get a low level of infection. That's a persistent infection. The other type is in the case of the herpes viruses, which, if you remember, their characteristic is that the herpes viruses are able to create latent infections. By latency, we mean that these viruses infect cells and then they can shut themselves down. So they can remain silent within these cells, hiding from the immune response, and then under certain stimuli, start to replicate again. And this can happen throughout your lifetime. And this is, as I described, the case of cold sores, where you get these so-called fever blisters in your mouth when you're stressed or get hormonal disruptions. And this can be itchy and quite painful. But after 10 days or so, that goes away. And what's happening is that the herpes virus creeps up your ganglia to go latent up there. And then when you get another stimulus, it comes down again. The same with chickenpox, another herpes virus, member of the herpes virus family. You, as a kid, some of you might have caught chickenpox, where you have this uncomfortable, itchy blisters all over your skin. After 2 weeks, you recover from the infection, and 30 or 40 years down the road, when you're middle-aged, you can develop shingles, where you have these blisters, these symmetrical blisters in the back. And that is your latent chickenpox that you got as a kid coming up again, reactivating. So these latent infections undergo these waves of what we call silent phases and then reactivation, which is reactivation due to various physical factors like UV light, stress hormones, even I believe cheese products, people who suffer from cold sores, fever blisters, various products in milk can cause these reactivations. So these are the major groupings of the basic virus pathogenic groups. So first we have the acute infections, where you get infection, disease, and then resolution. Then you have the persistent chronic infections where you get an acute, but then the virus gets controlled either through latency or the immune system. The third class of viruses are very unusual. These are called slow virus infections. These are, first of all, these are not due to what we know conventionally as viruses. They don't contain RNA or DNA covered by protein capsids with or without an envelope. These are single pockets of proteins, very similar to what causes Alzheimer's disease. So these are protein peptides which can get into your bloodstream and then get into the brain and cause polymerization, plaques and tangles, like what you get with Alzheimer's disease, to cause lesions in the brain. And that can result in dementia, paralysis, and so on. And this takes 50, 60 years once you get it. So this polymerization process takes very long. You guys weren't born then, but in the early 90s, one of the biggest scares in Europe, particularly the UK, was this so-called bovine spongiform encephalitis. And this is where various cattle farms in the UK, these cows were coming up where their back legs were totally paralyzed. They had to crawl along the ground. And what was really worrying was that their meat was being sold for hamburgers in McDonald's and all meat shops. And it was then figured out that the reason why they were getting this paralytic disease was that they were being fed with all mashed up products from other animals, which included vertebrae nerve cells. So the idea was that through this feed, this gross feed, they were taking in these so-called prions. These prions are these proteins which cause this neurological damage. So these prions are also the subject of intense research with sheep in a disease called scrapie. But again, research into this type of disease is very difficult because you infect sheep with this agent, the scrapie, and you'll only start getting symptoms 10 or 15 years after infection. So you can imagine how difficult it is to study this virus. So this is a summary of all these different patterns of disease causation in these different viruses. So we have the acute infection, like I say, where you have a burst in virus replication, pathology, asymptomatic or symptomatic. The immune system kicks in, and then the virus goes down. You get antibody protection, and you're immune. And this in the case of viruses like influenza or the hepatitis A virus. Then you have latent infections, as in the case of the herpes viruses, where you get an acute phase, the immune response kicks in, then the virus shuts itself down and goes latent. And months or years down the road, the virus might reactivate again to cause another acute phase. Persistent viruses or chronic infections, as in the case of hepatitis B or HIV, Again, you have an acute phase, and then the immune system kicks in, and then you have low-level, smoldering virus infection. And then these slow viruses, where somehow these prions get into the body and then do whatever they do, causing polymerization of the nerve cells. But this can take months or years before you start getting symptoms like paralysis or dementia or whatever. So those are the patterns of infection: acute, chronic, or persistent, slow, right? So that refers to the timeline and also is related to the virulence. Then we also divide patterns of infection of viruses depending on whether they can cross the placenta, whether they can damage a fetus at early stages of gestation. These are called what we call teratogenic viruses. So an example of this is another herpes virus called cytomegalovirus, which, when it infects an embryo in the first trimester, as that embryo is starting to develop all its different organs and everything, well, that virus is infecting these newly developed organ cells and blocking or disrupting the normal development. And so these babies are born with deformities like hydrocephaly, malfunctioned hearts, and so on. An example of this was recent, you might have remembered, about 5 years ago, the Zika virus, where particularly in Brazil, babies where the mothers were infected, the virus managed to cross the placenta and infect the babies, and these babies came out with very small, malformed heads. So these viruses can cross the placenta during gestation, during the first early trimesters. Another way that babies can acquire infections is through passage through the birth canal. And this is very prevalent with babies born to mothers that have chronic hepatitis B virus. That as they are born, they, through mixing of blood and whatever, as they pass through the birth canal, they can acquire the virus. This is one of the reasons why in certain countries they routinely give the vaccine to baby as soon as it's born if the mother is chronically infected with hepatitis B. And then for HIV, the virus that causes AIDS, one of the biggest successes in the past 20 years of bringing down the rates of HIV is by educating mothers with HIV not to breastfeed their children, because it was recognized that the virus could be passed on to newlyborns through breast milk. And then finally, we get to this group of viruses, which, like I said, can directly cause cancer. And this is because they contain various proteins within their genome that when they infect the cell, they can disrupt the signal transduction proteins, cell cycling proteins, or induce proliferation. These include viruses like what are called the papoviruses, SV40 adenovirus papillomavirus. The herpes viruses, particularly Epstein-Barr virus, which can infect B lymphocytes and cause immortalization, or in other words, that you can continually grow B cells infected with EBV in the laboratory and in the body. They can cause lymphomas. You have two human retroviruses, HLV1, HLV2. If you remember from yesterday, retroviruses are RNA viruses which can reverse transcribe back into DNA. Once they become the so-called DNA provirus, these can integrate into the host genome. And so you have two human retroviruses which can cause leukemias. And then hepatitis B and C, which is indirectly causing tumors through the immune system and the generation of mutations in these regenerating liver cells. Finally, we need to also consider how viruses are transmitted. So one of the most common ways is the so-called fecal-oral route. And this is particularly a reason why you have gastric diseases amongst young children in daycare centers, particularly viruses like papillomavirus, Coxsackie virus, rotavirus. These cause vomiting and diarrhea in young kids. In these aggregate daycare centers, play schools, where kids run around, don't wash their hands properly, constantly touching each other, shaking hands, they can pass the virus around. With the COVID-19, we now become more aware about respiratory viruses, which before, when we had influenza, we weren't really concerned about this. But we now understand this pattern between whether the virus infects the upper respiratory tract or the lower respiratory tract. If it infects the upper respiratory tract, it is less pathogenic, so it's less lethal, but it's more transmissible, because if you're infecting the upper respiratory tract, you're passing the virus around if you're sneezing or coughing. But then when it gets into the lungs, the alveoli, you have less chance of spreading it, but you have more chance of dying because you're getting pneumonia. And this is the pattern that we see with COVID-19. The viruses like the herpes virus are spread by direct contact or mixing of body fluids. So with herpes simplex, with Epstein-Barr virus, which is called the kissing disease, is transmitted more amongst young teenagers through lip-to-lip contact in terms of kissing. Other viruses can be spread, like HIV, through sexual contact. Then we have another group of viruses which are part of what are called zoonoses. Zoonoses refers to the fact that different animal vectors are involved. An example of this is the dengue virus, where, if you remember from your parasitology lectures like malaria, you have insect vectors that the virus replicates in these, and then the virus can then be passed on to humans when these insects like mosquitoes feed. This is in the case of yellow fever. Dengue virus is particularly prevalent here in Saudi Arabia, around the eastern province and Jeddah, from what I understand. Another common way of transmitting is through blood. So HIV, hepatitis B, hepatitis C. This is where a person who might be chronically infected and asymptomatic, then through mixing of blood, can pass that on to other people. The case happened a few years ago where a dentist in the US who had hepatitis C infected something like 20 patients. And so this is why, as healthcare workers, it's mandatory for all people who are involved in the medical field to be vaccinated against hepatitis B. Vertical. So this transmission from mother to fetus is what we call vertical transmission. Have you heard of this term before? So we have vertical transmission as opposed to horizontal transmission, which is from person to person. And then finally, to say something about diagnosis of the virus in the laboratory, we rarely do this because it is more for experimental settings. The easiest way, the fastest way to do this is to look for antibodies against the virus. But in many situations, you might not know what agent you're dealing with. You might not know what virus you're dealing with. So you first have to try to isolate and identify this agent. And one of the things you do is a blind test where you take blood samples from autopsies of people who died of, in this case, it was COVID-19 in Wuhan, and you blind inject that into animals, mice, rabbits, and you see if these animals get sick. Once they do, if they die, you take their blood and you try to see if you can see anything under the electron microscope. You do genetic testing like PCR because you have primers to various genes of different viruses. So you test these samples and you see whether anything comes up. Another way is also to treat or incubate these samples onto cells and see if those cells change behavior, if they show any what we call cytopathic effect, which would be due to the effects of the virus replicating in these cells. So in order to do this, when you're collecting blood samples from these patients, the same rules apply that you learned in the microbiology, that these samples have to be carefully contained in storage medium because you're carrying live biological samples. You have to protect yourself. You have to make sure that it doesn't leak, so you might be spraying it to someone, particularly in case of very dangerous viruses like Ebola, Marburg, and so on. You have to make sure that they're stored at proper temperatures, that they don't get too hot because they're going to kill off any of the biological material, and so on. So, as I said, there's different ways of trying to identify what the agent is. One is through isolation. The other is to detect through PCR, antiviral RNA or DNA, and then to look for antibodies. Why our knowledge of viruses is a lot more limited compared to bacteria is because of the fact that we have to grow the viruses in cells. And viruses, again, because of this term tropism, that depending on the virus, they can only grow in particular types of cells, liver cells, kidney cells, lung fibroblasts, neural cells. So if you don't know the properties of the virus, you have to test this virus against all of these different cell types to see if it'll replicate successfully. And if it does replicate, then depending on the virus, depending on the cell, you will see these so-called cytopathic effects. So these are virus-specific changes, phenotypic changes to the infected cell. And these changes can be in the form of these, what are called nuclear inclusion bodies. So within the nucleus of these cells, you can see these big clumps of material, which represent viral proteins being made. Viruses can also change the cytoplasm of the cell it's infected by causing these vacuoles to appear. Many of these vacuoles can contain fat bodies. Some viruses can cause the membranes of cells to fuse with each other, to cause these so-called multinucleated giant bodies. So this is one cell, which is just a fusion of all of these different cells together. And then other viruses can cause cell lysis, where you get these gaps in this typical cell monolayer, which represents one virus that's infected all the nearby cells to cause these islands. This is a basis for quantifying how much viruses we have in a blood sample, because based on this idea that one virus particle will cause one infected cell to infect all the other cells around it, to create this gap in this monolayer, which we call a plaque. So what we can do then is to make dilutions of, if we're trying to compare blood samples from two different people to see who has the biggest number of viruses, we will dilute those blood samples and do a limiting dilution assay on live cells, and then count the number of these so-called plaques, just like you learned in the immunology practical with the ELISA test, right? Where, if you remember, when you are, say, measuring antibody concentrations between two different samples, you dilute out those samples, and the ones that have the highest concentration of antibodies will be diluted the furthest. At the end of that dilution is beyond which you won't see any reaction. Similarly, the more virus you have, the more dilutions you have to make before you don't see any plaques. So the blood sample or whatever that has the highest number of virus particles will be diluted much further before you don't see a reaction. So this is the basis of the so-called Plaque Assay. And then we can try to see if we can identify the viruses by electron microscopy or by using fluorescent labeled antibodies to particular virus proteins. And so under the UV microscope, these are infected cells, and these spots represent the virus proteins in the cytoplasm. And then the more reliable, which is what is being used currently with COVID-19, is to do PCR, where you use primers specific for particular genes in the virus, and then you can pick them up as bands. Yeah, this is what I was describing in terms of the limiting dilution assay. Okay, that's it. So the question is, do we need to know which family the virus belongs to? For example, cytomegalovirus and chickenpox or herpes viruses. Yes, you do for these common viruses, right? So herpes virus is a particularly important group of viruses. So at this stage, you need to know what herpes viruses are. Epstein-Barr virus, cytomegalovirus, herpes simplex, varicella zoster. What are the common viruses we need to know? I'll tell you that in the next lecture. Okay. All right. Thank you.
Are you reading the case study?
Yeah. Okay.